Hello, and welcome to the Rosa Worldwide Webinar Series. My name is Mike Reed. I'm the Chief Scientist at Rosa, and I'm happy to welcome you to today's webinar. I'll just note that during the presentation, you can enter questions using the question and answer box. There's a button at the bottom of the screen you can click to access it. We'll also be sending out email announcements for upcoming webinars, so please check your email or the Rosa website for those announcements. There'll also be a link in the chat window for a survey that Rosa is conducting on the state of QSP in the industry. It's purely optional if you'd like to answer it. So at this point, I'd like to welcome today's speaker. Today's speaker is Damian. Valerie is a QSP and PBPK consultant with 26 years of experience, most recently at GSK, where he led the systems modeling and translational biology department, overseeing the application of QSP and PBPK from discovery through development. A recognized leader in his field, Valerie has held influential positions in ISOP and ASCPT, championing the advancement of QSP science. He's made significant contributions to QSP model development and tools, as well as PBPK modeling for inhaled, transdermal, ocular, and long-acting injectable delivery systems. Welcome, Valerie, and we're looking forward to your presentation today.
Thank you very much, Mike, and thanks for the invitation. I'll turn my video off. Hopefully this is going to make things a little bit easier. So thanks for the invitation. This is basically a good time to actually revisit the presentation that I've done in this webinar in 2017, where I was talking about, well, QSP, what's in it for me, trying to justify, if you want, how QSP can be useful for various leaders within pharma companies. A lot has happened in the last 7 years here. There is significant growth and maturity of the QSP field. There are a lot more applications across discovery and development. There are many more CROs that are specializing in QSP within different pharma companies. There, the shift has been from lone modelers to dedicated QSP groups. Many published QSP models out there, and many QSP models are now available for licensing from the various vendors out there. There's very clearly defined QSP workflows, and I'll touch upon that shortly. And there is significant regulatory interest, and that was enhanced now with the opening of the Quantitative Medicine Center of Excellence by the US FDA. So the agenda for today is I will talk first about what does it mean, why is it must-have for me in terms of applications of QSP, talk about the quantitative medicine toolbox, couple of impact examples, mostly from my GSK tenure, about QSP workflows, 2 examples on digital twins and target pharmacology assessment, and with some challenges and opportunities. So let me start with, why QSP is a must-have for me. And this is actually a slide that was put together by Paolo Vicini and Pete van der Graaf in 2013, and articulates very beautifully how, in order to have confidence in proof of concepts, you need to have confidence in compound and confidence in target, with the confidence in target being sort of the core of what QSP is all about. I wish, though, that he would have changed the order of these two, because for me, we should start with confidence in target before the confidence in compound. The trying to check if a novel target works in human takes 5 to 7 years of discovery, takes hundreds of millions. Doing the same thing using QSP, it's actually about 10 times faster, give or take, and easily 100 times cheaper. So this is basically, from my perspective, the place where QSP has a huge role. But this is me. Let's look at what else does this mean? It's not just myself. For this, let's look at any kind of discovery and development effort in pharma. Try to establish a relationship between dose, drug in plasma, drug at target, target engagement, pathway modulation, cell and tissue level effects, and outcomes, clinical outcomes, and biomarkers. The dose and the dose in plasma, and as well as the outcome, are at the organism population level. The drug at target and cell tissue are obviously at the tissue level, and target engagement and pathway are at the molecular level. You can see already that now you have a couple of disciplines that are involved in making these kind of connections. At the top, we have the clinical pharmacology scientist or leader, and this is basically one of the types of individuals towards the QSP is a must-have. They link the with the clinical, and in doing so takes likely a month. At the tissue level, you are looking at preclinical leader, the MPK safety. At the molecular level, you are looking at biology and looking at chemistry leaders. And in addition to this, if I look at all of these individuals within pharma, we're leaving out two other leaders, if you want, that are involved: the regulatory and the patients. So I'm going to try to look at all of this from all of these points of views. So coming back to this diagram that shows the quantitative medicine toolbox, in order to go and establish relationships between drug in plasma, drug at target, all the way to the clinical outcomes, there are three different approaches: either do an experimental design or a clinical trial, you do an empirical modeling, or mechanistic or knowledge-based modeling. And let's see what these look like. So you can do a clinical trial. Obviously, that links the dose to the clinical outcome. You can do a PK study that links the dose with a drug in plasma or drug at target. You can also measure potency that, but perhaps in vitro, that links the drug at target with the target engagement, or you can do it in vivo. The drug at target connected to the cell and tissue level is basically a cell-based assay. So these are choices that one has if you are looking at establishing experimentally links between these different steps. If we look at empirical means that you actually have the data, and then the simple model that explains that particular data that you can use for simple interpolation. That okay, the drug in plasma, drug and target or in plasma with the clinical outcomes. But then when you start looking into the mechanistic realm, you have the physiological based PK that links the dose with the drug in plasma and drug at target. You have the molecular dynamics or free energy perturbation type calculations that by mechanistically to figure out the binding to the target with the effects at the pathway level. And then you have QSP that links what's going on at the pathway and the target engagement level with the outcomes. So effectively, if you are looking at the top row, these are experimental is usually costly to generate. If there is no other way to demonstrate understanding the definitive answer. The empirical are easy to build models, easier to defend, but in order to build a PK model, for instance, you require the PK data. In order to build a PK/PD model, you require a clinical trial. So it's require costly data and is limited in extrapolation. The mechanistic, on the other hand, is much more challenging to build and defend. It can rely on in vitro data if need to, and it does provide a lot understanding and allows for extrapolation. Now, let me bring another item to this. We talked about experimental, mechanistic, and empirical. Well, there is the artificial intelligence and machine learning. I'll have more about this a bit later on. As it stands now, first of all, it can handle a lot of these, obviously not the experimental part, but it handles a lot of these predictions. It can improve workflows at all the different levels, and it is easy to use. Just as a joke there, just chat GPT and it's going to give the models that can hallucinate, that is as salt and pepper. You are putting salt and pepper on almost any dish, and it makes it taste better.
Hey,
But you
Valerie,
need to
would you
put
mind just
just the right
checking your
amount. You put
microphone and
and
making
rely
sure
too
that
much
it's close to
on.
your mouth? Thank you.
Okay, I'm gonna try to speak a little louder.
Thank you. Appreciate it.
Okay, all right, that should work. All right, so this is basically the toolbox, but let's look at how this toolbox now looks like when we talk about QSP during discovery stages. So when I'm talking about discovery in terms of, during target validation, at that point, we don't get a drug to the potency and the target your problem. So you have to rely on mechanistic modeling, QSP in particular, to actually make the connection to the clinical outcomes. And the questions that can be addressed at this stage are things like maximum achievable efficacy, target engagement required for efficacy, balance between potency and let's look now what's happening during the translation phase. In the translation phase, you already have a particular molecule. You haven't tried it in humans. You have animal data, you have in vitro data. Therefore, you can use PBPK type model, mechanistic model to understand what the drug is going to look like in plasma and in the target through leveraging PBPK approaches. By that time, probably you've measured potency both in vitro and cell-based assays. And you connect that to a QSP to provide a QSP and PBPK, of course, to provide the more accurate dose estimates, balance efficacy and safety, look at different dosing regimens, combination versus sequential, look at
Excuse me, Valerie.
biomarkers.
Excuse me, Valerie. We're still having issues with your audio.
And
would you mind just trying to close any other applications you may have on your computer? It might be a bandwidth issue. But your audio is breaking up for some listeners.
Let me see. Let me try this. Is this any better?
Yes, that sounds better. Thank you. I think it might have just been an internet bandwidth issue.
Okay.
Thank you.
Apologies about this. All right. So moving forward, if we move from the translational phase, the next step here would be to look into the clinical development after first time in human. During first time in human, obviously you have experimental data for the PK, and therefore you can afford to do now to replace the PBPK model with the actual PK for predicting drug in plasma. And
moving forward, you still have to use the cell-based assays and the QSP to try to predict what's happening in the clinic from a PD perspective, and that allows you to do a much better and more accurate dose prediction, predicting exposure, help design the clinical trials, look at different patient population, and so forth. Moving forward, we're changing to late-stage clinical trial development, and at this point, we have experimental data on the PK and the clinical trials. You have experimental data for all of this. Therefore, you can afford to use a simpler empirical-based connection from a modeling standpoint. So a PK, PD, a pop PK type work is actually quite useful. Of course, you can still leverage PBPK for drug-drug interactions, special populations, peaks, setting manufacturing specification, and of course you can still use QSP if required for extrapolation. And one quote that I had from a presentation for Pete Van when the answer to the question is within the clinical, looked at QSP application for me, validation and preclinical development, clinical development with the appropriate leaders in each stage. Moving next, look at the regulatory agencies, which actually are quite keen on advancing the science, accelerating the decision making, the regulatory science. Demonstrated through the creation of medicine center of excellence. Next is the portfolio decision makers. These are the pharma executives. There is a lot more that we can do for them. This is managing risk at the portfolio level, prioritize programs, balance the portfolio. In licensing decisions, a few, not enough cases where QSP model is used for making a licensing decision. And finally, patients. I think there is a lot to be said about using QSP to provide personalized treatments. I'm going to be talking more about twin approaches and also about rare diseases. So with all of this, this is applications of QSP that have grown quite a bit in the last years. How much? And one way of looking at this is to look at this publication from the US FDA that basically shows 157 applications in the last, I think it is 10 years or so, and you see very clearly that the trend is it's going upwards. And moreover, there is this very nice from an FDA industry scientific exchange program where criteria are needed for credibility, for validation of QSP models, and how does that balance with the risk going from internal decisions all the way to phase 1 and phase 3 and labeling. What's interesting about all of this is that there is a huge amount of application of QSP in this internal decision steps that, of course, you'd like to have a huge amount of validation for it. But in a pinch, you can actually get by with a little bit less validation than what you would normally require for influencing labels. I want to start talking a little bit about examples from my previous experience. And this is one model that on acne, and I'm not going to talk about the details of the model per se. I've talked about it in several other forums. The model is shown down below in terms of what's included in this. What I'm focusing on here is basically the life of the platform model. And we started development actually with Rosa in 2013. We completed the first version of the model. We use it to deprioritize the clinical trials due to minimal differentiation. we basically went ahead and tried to select optimal candidates. So what you see in this diagram here, you're looking at lesion severity of the standard of care treatments. You want to basically bring this to clear skin all the way to the bottom. And then you see here different compounds with different mode of action, different targets. And you see as you increase the drug concentration at the target site, the efficacy after dosing starts to, the clinical outcome starts to decrease towards clear skin. And you see here that, for instance, with this compound Z, it's actually not possible to get efficacy better than what's already there on the available on the market. So no benefits to patient, whereas for these two, the one in green and blue, there is this possibility if we manage to get the right concentration at the target site. We integrated the QSP with PBPK. In PBPK, added additional target dates. So we basically been using the QSP model in several places to help the various programs. We repurposed part of the model for different indications. And then about 5 years later, you can look at the time there, this 18, the use of the model, had the need to actually add new biology, new targets, seek new patient phenotypes, do a lot more work in developing virtual population, and again, support with this new model several new discovery projects. Another example, this is looking at the complement pathway. And you can see here the diagram of the model. It's actually, you can see the model in terms of different engaging different one are feasible and which are not. If I look in the timelines here, in 2014, we basically started with building this model, and it was just a suspicion that the target that we were interested in at the time might not be ideal. The model was not complete, so we didn't have this to actually take decisions based on it at stage. We started to develop this model. We evaluated the targets, and the conclusion held that that particular target might not be ideal. So the project team was, well, the model must be wrong because you are not. There were a couple of experimental data sets that we were not able to reproduce with the model. Well, we integrated the QSP with PK model for those predictions. We designed novel experiments in there. And then when all of those experiments actually, the new experimental data and the model sort of were in sync, things have changed. And it was a very significant change in the program team to a predict first culture, where any kind of new targets, modality pairs they were interested in pursuing, they were coming to us, to the QSP, to provide that. We looked at virtual patient simulations, looking at response variability. We looked at designing experiments for evaluating new targets. So again, you can see, just like as in the acne model, quite a long life for this QSP model. Even though the initial development was quite significant, it actually had a utility over long term and over many programs. Finally, the last example I wanted to give here is on the oncology model. Using a modular approach, a submodel that describes all of the interactions for that particular cell type. This diagram shows just interact at the level of cell-cell interactions. Simplicity, the kind were not included in here. And this model was put together around 2018. Different stages in developing the model, including a new targets, costimulatory receptor A and B. In the beginning of 2019, we calibrated the model to the healthy states, calibrated to cancer patients, and then we started to truly have significant impact. This is evaluating mono and combo therapy for the two targets we've included, added the third target in here, evaluating the effects of these. And then from 2020 to 2024, we've started implementing a host of other new IO targets. We basically had a very extensive virtual population generation to match outcomes and distributions. We took the model and recalibrated for different cancer indications. And with all of this in place, we guided development for a couple of programs. We contributed to early termination of discovery program due to model predicting lack of efficacy, both as a first-line and second-line treatment, and including successful submissions to the FDA. So I'm going to move now and sort of look at the program questions addressed by QSP in 1 year. This was actually in 2022. And you can see here on the discovery side, level of target engagement required for efficacy. This was done for 10 programs within that year. Best modality, mode of action for a target, evaluating combinations, maximum available efficacy by the standard of care, balance between ADME and efficacy, and so forth. And then on the clinical side, we can look at combination evaluation, again, comparison with standard of care, dose and regimen optimization, patient stratification, biomarker evaluation, and treatment positioning. Quite a number, and this is basically work done within the whole team during the course of 1 year. So I'm going to switch gears slightly and start talking about modeling workflows. And this is a that I put together and presented actually at the Rosa webinar in 2017, where I was trying to make the case that when you want to build a model, you put some effort in developing a QSP model, and you get some benefit from doing so. If you look at all of these and sort of put there are quite a number of publications that actually describe QSP workflows. The QSP workflows in more recent publications talk about defining the model scope, building a reference patient, define virtual patients, cohort, defining and validating a virtual population, and then you start answering questions. If I put the colors here on the diagram, there is one thing that becomes very clear. We spend a lot of time in the beginning, and this again looks at 7-8 years ago, on building the model and building the reference patients and validating that, and a lot less on looking at these two pieces, calibrating and validating a virtual patient cohort and virtual population. Therefore, in what looking more accurately and how we spend time today, well, there is a lot more that's done into the validation, and this is partly because of that discussion that we just had on the from the US FDA, the effort that needs to be put into validation of these models. So the workflow essentially is you start with defining the intervention, gathering the data, defining the outputs, separate calibration, validation data, and you define the model, calibrate the parameters, and then you start talking about virtual patients. And virtual patient is essentially a parameterization. How do you determine which parameters to define for the virtual for each of the virtual patients? And you do this by doing sensitivity analysis. You generate virtual patients, select plausible virtual patients, calculate a virtual population to match the calibration data, and then you validate that everything is correct. In the next slide, I'm going to be talking very briefly about a few of the challenges in any one of these steps. Selecting the parameters of the model that characterize a virtual patient, you can do it through an expert opinion, local sensitivity. These currently almost always is done through using global sensitivity analysis, and there are many algorithms for these ones. And there is obviously a challenge because all of this is very, very computational intensive, and you are hitting the curse of dimensionality. The second one is generating the VP and the plausible VP. And the issue here is not generating the VP. You can sample the parameter space quite easily, but in order to check that this a patient is actually viable, you need to essentially, quote unquote, enroll it in various trials. And that is quite computationally expensive. And trying to verify that these patients give the right response in all of the trials, in our practice, we've seen less than 1% of the virtual patients that we generate are actually plausible. And in our experience, it's just 1%, maybe the best we've seen is about 5%. So that needs to be improved, trying to use surrogates or other approaches or machine learning there to improve this ratio. And finally, in order to do the calibration, we need to look at a prevalence weighting. There are quite a few Monte Carlo sampling approaches, MCMC type approaches, to actually generate virtual population to validate to calibrate and validate the data to the distribution of biomarkers. This is just, I'll skip very quickly, leave it like this for just a second. This is looking at there are quite a number of publications that describe workflows all the way from 2007 to 2023, including several from the US FDA. I'm going to switch gears and talk a little bit about the digital twin application. So the definition of a digital twin from our trusty Wikipedia, a digital twin, it's a digital model of an real-world product, system, or process that serves as the effectively indistinguishable digital counterpart for practical purposes. All right, so what does this mean? It means that if I need to define a digital twin, I need to define two things. I need to specify the system or the process that I'm creating a digital twin for, and the purpose. Why am I doing this? So let's look at some examples. Let's say I want to create a digital twin for a disease X. Why do I do it? Because I want to predict the disease X population response to therapy. Well, that's exactly a QSP model for disease X. How about if I look at building a digital twin for drug I? Why? So this is basically the purpose is predicting population exposure for the drug. That's effectively PBPK for drug Y. How about if I look at the system as being patient Z and drug Y? Basically for the purpose of predicting individual exposure for patient Z to the drug Y. And that's effectively the PBPK parameterized for that particular patient Z. And finally, the one that probably is most relevant to this audience, looking at a digital twin for patient Z and disease X, predicting individual response to therapy for that patient. And that's effectively a QSP model for the disease, and parameterized to match the response of that particular patient. So with this in mind, a digital twin for patient Z and disease X is a virtual patient, but not all virtual patients are digital twins. All right, so looking at the definition further, the digital twin is a set of adaptive models that emulate the behavior of a physical system, getting real-time data to update itself along its life cycle. So a digital twin is the best that can be done with available data for the system, and it adapts when new data is available. So I'm going to give you an example here. This is a work that I've done in collaboration with UCSF, looking at bone marrow transplant. And in bone marrow transplant, you basically have a patient, they undergo chemotherapy, they have the transplant, you follow the neutrophils for 20 days after transplant. So in this context, I want to build a digital twin for the bone marrow transplant for the purpose of predicting population neutropenic state. Well, that's effectively a QSP model for bone marrow transplant. But what I want to look at the system and have the I want to build a digital twin for a particular patient Z undergoing bone marrow transplant. And I want to build that because I would like to identify the patient response to administer optimal rescue treatment. This is obviously a parametrization, but it begs a question: how much data do I need to know about patient Z to build a proper digital twin? And this is basically looking at, in this particular case, you have start of treatment, you have end of treatment, more data coming through. As you incorporate more data about a particular patient, the digital twin accuracy increases. So you start here with a generic digital twin. You just look at leveraging population availability, population knowledge about what may happen in this particular case. You have pre-treatment digital twin, where you can look at the individual's medical history and try to predict what's going on. On the other side of it, you can say this is the true digital twin, very accurate. But let's look at the usefulness for the patient, because if I plot again on the same kind of a diagram, the usefulness in the beginning of the treatment, it's going to be very useful if you can predict the patient response before starting the treatment. But if you have a perfect model at the treatment end, the model, the digital twin, it's useless. It's too late to make any meaningful decisions afterwards. So the idea is to actually look at what is a useful the digital twin somewhere in the middle. So the model that we put together is based on neutropenia models. There's quite a few out there. There's basically the host proliferating cells, couple of transit stages contributed to the neutrophils in the circulation, the GCSF as a feedback mechanism. This is for the host individual. The donor cells are contributing in the same way. So you put the donor cell, they have the same kind of processes that they undergo. And obviously the neutrophils in the blood is the sum of the two. Now, these patients undergo chemo. So we've built for the team a very simple model, a generic model for this. And the chemotherapy, depending on which type of chemotherapy is, can either inhibit the proliferation of the host proliferating cells, or can kill the cells, or can prevent them moving from in this transitional stages. Now, we can look at GCSF effects based on modeling the details of the GCSF receptors. And again, they have access, they influence the transition from one stage to the other. Host immune system. So I define the host immune system as the number of neutrophils that are available or being proportional to it. And it is that host immune system that effectively kills some of the donor cells, and vice versa. And finally, the key drug that's given to actually free up the space in the bone marrow for the donor cells, that's busulfan. Busulfan has a very tricky PK, so you basically have the PK with the clearance as a function of body weight, age, and so forth. And then we accounted for a direct effect and then a delayed effect of busulfan. And you'll see them, the direct effect on the transitional stages, and the delayed effect on the killing of the various cell types. This is the model that was put together, and just to give a sense about the overall, how the data looks like, quite huge variability, because every patient here is different age, pre-existing conditions, chemo treatments, and rescue medication is quite different. When you're looking at how well we're doing, and let's just focus on one particular type of patients, you can see that this is the error that you're getting if you just build a digital for the disease. You can improve on that if you are looking at digital twin in this particular population. So you do a population a pool fit, if you, and if you're doing it individual fit, of course, you're going to do digital twin for each patient, leverage all the data you as well. This is looking at patient level data. So this is these different patients. This is the protocol when the different rescue medication was given. And you see here the useful digital twin. This is the day of the transplant, day 0, and you see a much wider distribution of the prediction. But this is the data that was used to calibrate the model was up to day 7, and from here on is prediction. Here you are looking at using the full data set, and you see a much tighter prediction at the very end, but obviously the usefulness here is going to be much lower. So how do we figure out what is the right balance? So we looked at trying to see if I just use the data up to date, let's say 7 or 14 or 21 and so forth, and try to predict look at the prediction error, you have effectively the pre-treatment digital twin that doesn't do particularly in the end. They all do very well in the beginning. You know, this is the time when the neutral count is going to get quite low. And then you're looking at the optimal, which does reasonably well. So I'm going to skip in the interest of time, and I was looking at I'll just mention it very briefly. This is actually target pharmacology assessment. It's another way of looking at applying QSP in very early discovery. You can think about it as being it's a P-like chemistry, and you can afford to build this actually very early in discovery to identify which are the best molecules to develop. What you get out of doing this kind of analysis is level of target engagement, the drivers for efficacy, modality comparison, prioritized series of compounds. And what you need to put in is a model for the target pharmacology, target engagement and modality model, and the PBP model. And the way that this works, and there's a couple of publications around this, is that first you need to build a PD model. Then you need to add the ADME properties to it, and generate virtual compounds that you are scanning. Then you are looking at predicted PK and PD profiles. And then you finally put all of this together with machine learning to actually identify a decision tree, look at endpoints that drive efficacy. I'm going to skip the actual examples because it's in the interest of time here. And just about challenges and opportunities, and try to focus particularly on artificial intelligence and machine learning. So if I look at data and knowledge that we're using in modeling, it falls in the 3 major classes. First is targeted data, which is high precision data like measurements, PK or body. It's focused on the most relevant measurements. It's big data. It's basically omics type data, databases, sales data, and so forth. It's comprehensive, it's structured. And then there's big knowledge in the middle, which is basically published data knowledge, known physics, chemistry, biology, basis, and peer review. This captures the knowledge of what's going on. So what kind of models do we do for each of these 3 types of data? So for the big data, one does machine learning type modeling, data modeling. For the knowledge, you're using mechanistic modeling, QSP or PBK. And for targeted data, it's pharmacometrics type modeling. The machine learning part gives you a correlation and gives you a hypothesis to try. I've been talking with folks in machine learning, and they're basically saying, no, we can try to get that causality, and there is effort in that space. But for the time being, correlation is kind of the main thing that machine learning is doing. This is looking at and providing understanding, causality, and confidence, and then this is looking at evidence. So you can go from correlation to understanding to to evidence. This is data first, this is model first, and this is experiment first. So this basically brings me to the point of we need to connect now the machine learning with QSP. And the way I've looked at it is, why would we want to do that? And the first thing is, well, we want to do things faster. We want to build a model faster. We want to use natural language processing to actually infer the structure of the model. Or maybe, and this is much more available these days, to code faster. Once you have a model in place, you can much easier generate code to do the analysis. You want to run the model faster, do the virtual patient generation. Again, that can be done with using machine learning by using surrogates or model reduction algorithms. There's quite a number of them in that space. Identify valid virtual patients, inverse surrogates, and population calibration. Again, there are quite a number of advanced patient interfaces approaches that can be used in there. The next reason why we may want machine learning is we want to do something novel. We want to identify novel correlation between VP patients, novel diagnostics, and this is actually quite a lot of work that can be used using perhaps traditional machine learning approaches on virtual patients and be able to predict responders. But I think that the interesting bit is looking at the last part. We want the AI/ML to do things better. Replace the ODE model perhaps with mixed ODE and machine learning. A neural ODE, it's an example of that. The physics-informed neural networks and so forth. There's quite a number of approaches out there, or integrate mechanistic models with omics data. And this actually would be a very good way to link to digital twin technologies. So with this, are we there yet? This is a slide that I pulled actually from a conference presentation in 2012 that basically claimed that Apple iPad 2 is as fast as the Cray 2 supercomputing. And I had this slide actually to present even in 2007. And I was looking at complex QSP modeling today as being the same messy environment as Cray 1 was in the 1970s. Perhaps we moved away from that stage. With all the workflows, we're not quite in the 70s. We're probably in the 90s or so. But are we here yet? Are we basically using the QSP model as simple as just using an iPad? And I don't think so. We still have some ways to go. It took for designing these systems, about close to 35 years. We are probably around 12 or so, maybe in development of the QSP. So we are still there. We still have some ways to go until the use of QSP becomes as easy as just simply playing with on a tablet, on iPad. And one of the biggest challenges that we have these days in terms of promoting QSP and getting QSP to be used more broadly is basically we are requiring quite a bit out of QSP scientists. We want them to know mathematics, biology, clinical, statistics, communication, programming. There is quite a number of skills and quite a few that are being taught at this point in dedicated QSP programs in academia. But nonetheless, there are at this point a growing number of QSP interested individuals in academia, and obviously quite a demand for high-skilled individuals across the industry. I would end with the acknowledgements. There is quite a number of folks, particularly my colleagues from GSK, that contributed to a lot of the impact slides that I've described. Folks in the systems modeling translational biology, folks in GSK dermatology, in VIV, in immunoinflammation, the immuno-oncology and respiratory teams, and collaborators. Rosa was one of the first that we worked on developing QSP models in SysBio. UCSF, this is the example on neutropenia, and Clemson was involved in the immuno-oncology in the beginning. And with this, I'll stop here, and I'm willing to entertain any questions you have.
Thank you, Valeru. To everybody on the line, apologies for the audio issues, but this was an excellent presentation. So we have just a couple of questions here. First one is there a risk of extinction of QSP groups within pharma companies? So these are the ones that are, FTEs embedded within the organization, perhaps due to the influence of CROs, the progress of AI, or for other reasons. So do you think, this is speculation, do you think QSP groups within pharma companies will grow or shrink in the future?
I hope the answer would be that they will grow. I think that there is an increasing interest from regulatory in having QSP models, increased realization in pharma companies that QSP is the way to go. But as with everything, whenever it comes to having more QSP models done in a pharma company, it's also possible that there is organizational issues. And it's not that QSP disappears, it's just that it gets reorganized and spread in different parts of the organization. What I've seen is actually not as much that it's you go from a centralized group to actually having groups dedicated to building QSP models for a particular therapy area. But if you look overall, I don't think that the number of QSP modelers and the demand for QSP is going to be going down.
So on a related topic, what are some strategies that an internal group or a consulting group can use to increase adoption and acceptance of QSP models within an organization? How do you make the case for using these to the rest of the organization?
So I think that there are a couple of ways. One is looking at case studies, applications. It's quite easy, if you want, at this point. It wasn't 7 or 8 years ago to find applications of QSP and what can be done through what the impact of those applications was. So that's one way that you can convince people that QSP is the right way. The other one actually is quite an interesting one, and I'll give an example there. Like if you want an analogy, I participated in an FDA workshop maybe 10-15 years ago on PBPK, QSP on PBPK. And at the time, a question arose from the audience about how often have we seen, I don't know, it was a question about how often have we seen predictions within some fold in all of the PBPK model received by the FDA. At the end of the session, there was a half an hour break, and at the beginning of the next session, the answer was there. And that blew me away. That particular
I'm sorry, can you repeat the answer? I think we missed the end of your comment. If you could just repeat that.
Internally, within in about half an hour, the answer came back from the US FDA about how often that particular was found, that error was found in all of the different submissions, and that blew me way because it meant that within a half an hour, they had the tools available to scan, to query all of the submissions and find that particular question about frequency of that error. And I use that with different to say, well, if we don't build those kind of models with the company, the regulators, they're going to basically look at our submissions. Things would be flagged that we would be even aware that they're there. In actually building. That's the PBPK example. However, with all the publications that have come more recently on the QSP, I start with the starting of the quantitative medicine center of excellence, FDA. So that's not an argument that you can make to convince people to invest in.
Okay. Another question, how does machine learning deal with ODEs, such as are used in QSP?
That's a very interesting. Machine learning, there was actually a fantastic, maybe about 3 years ago, that basically demonstrated that the way that
I'm sorry, we're still losing your audio, Valerie. I think at this point, we do have a couple of questions from the audience, and perhaps, Valerie, you can follow up by email after the webinar. That might be the best way to answer some of these. There's one or two specific technical questions as well as more global strategy questions. But
Okay.
At this point, I think I'd like to thank the speaker and the audience for attending today. Please keep an eye out for upcoming announcements for upcoming webinars from the Rosa Worldwide Webinar Series. And I thank you for joining us today, and I thank you, Valerie, for speaking with us today.
Thank you. Thanks for the invitation.
Great. Thanks,
and
everybody.
Apologies for the connection.
Yep.
Features.
All right. We'll see you next time.
Thank you.